Hi, this is Miss Litton, and this is my wonderful, excellent Period 1 AP biologist. Say hi. Hi. It's a special one we have. This is chapter 8, cellular respiration. We've already done photosynthesis, and we're very thankful for photosynthesis because what are the products of photosynthesis that we appreciate? Oxygen and glucose. Good. Okay. So we can, that's not us, but we can eat and we can breathe. Now, who does cellular respiration? Good. It happens in the mitochondria, but what kind of organisms do cellular respiration? All cells. Yeah. All cells are going to do cellular respiration because cellular respiration is a way of cashing in our glucose, which is like a $100 bill, for some ATP. Okay? The energy currency of our bodies. So this reaction you already know. And I would like to have the youngest bio buddy just review this a little bit. We've already talked about it 2 chapters ago. Go ahead.
Like ATP, they separate ATP, and then they all go together and they form ATP.
So on your basics, you break down carbohydrates. You're breaking down carbohydrates in order to build up what molecule? ATP. Good. To build ATP, it consumes what gas? Oxygen. It produces what gas? Carbon dioxide. Good. And then I gave you the equation right there. And we say it happens in land and water. If you need oxygen, are the fish taking the oxygen out of a water molecule? Are they splitting water in order to get their oxygen? No. Oxygen is dissolved in water just like you dissolve sugar into coffee. Okay, so I don't want to hear that I don't want somebody to think that they're splitting the water in here in order to access oxygen. Oxygen is dissolved in the water. What kind of water do you think has the most oxygen? Fast-moving water or slow-moving water? Fast, because it's getting the oxygen mixed into that water. Okay, when you have slow-moving water, that can be a problem and get depleted of that oxygen because it's not getting mixed in there. Okay, now we've already talked about oxidize and reduce. Youngest bio buddy, you just went, right? Yep. Oldest bio buddy, you explained this equation right here. You should have no problem.
Okay, so pretty much what
All right, let's take a look at your notes. The high energy molecule we are using, this potential energy is called what? What's the high energy molecule? What is it? Glucose, good. Low energy, CO2 and water. So what's happening to the entropy? Remember we talked about this? If we're breaking it down, we're going to greater what? Entropy. Good. Okay. We have an electron pathway again, and here's a little summary. Glucose is going to get what? Oxidized. So is it gaining or losing energy? Losing energy. It's getting oxidized. The energy is getting released out of it. Electrons pass to coenzymes, and now instead of NADP, it's just plain old what? NAD, and there's another one called FAD. We have a new playa. Which then drop the electrons off at a what? Electron transport chain, which ultimately will reduce oxygen into water. Energy harvest from an electron transport chain. We've already done this before. Whenever we na-na-na-na-na-na, what can we make? What is that called? Make some ATP. Good. The reactions of cellular respiration allow energy and glucose to be released slowly. Therefore, ATP is produced gradually, still only about 39% efficient. All right, now the coenzymes. We used NADP before. Now our coenzyme is just NAD. Here on the bottom, They've drawn it out for you. There are some things that you should recognize here. Here's adenine down here. What is adenine? It's a base. Good. Okay? And you have sugar and phosphates in here. Okay, so it is associated with like a nucleotide, what we're already familiar with. So I have a little video I'm going to show you here, but I need to pause this due to copyright. Okay, welcome back. So on your notes, go to where it says coenzymes, NAD and FAD, each which can accept electrons temporarily to form reduced NAD and reduced FAD. They are not consumed. They are recycled and reused. They are not consumed. They are recycled and reused. Okay, so when we did photosynthesis, we had two basic reactions. We had the light-dependent reaction and the light, what? Independent reaction. When we do cellular respiration, we are slowly releasing the energy in many small steps, and the majority of that energy is getting stored as reduced NAD, which we will then take to the bank of the ETC and cash out for some ATP, okay? That's where we're headed. So the first step is glycolysis, and let me get all your eyes up here because I'm giving you the big overview right now, okay? The first step is glycolysis, and I want you to know that glycolysis occurs by all cells. All cells do glycolysis. You don't have to have mitochondria to do glycolysis. This happens in the cytosol. It's like a 10-step reaction. You're not memorizing all 10 steps. Just telling you the beginning and the end. A 10-step reaction which will harvest you a net of 2 ATP. No electron transport chain is used. Okay? It is a substrate level phosphorylation, and I will explain that. You're not using electron transport chain. It's basically, I have the phosphate, now you have the phosphate. Okay? And when you are done with glycolysis, you will walk away with 2 ATP. That's not enough for us to survive on. We need to further take the energy out. So we continue on and we do what's called a preparatory or transition reaction, which brings you into the mighty mitochondria. And here in the center of the mitochondria is called a matrix. That will be the third step, called the citric acid cycle. Okay. Another name for it is the Krebs cycle, and that's named after the guy, just like the other one we learned about was the Calvin cycle. It's named after the person who discovered all the intermediates. And then we do the electron transport chain and the chemiosmotic synthesis of ATP. If you do this, do you remember before when we did who caught the electrons at the end of photosystem 1? Who caught the electrons? NADP forming what? Reduced NADP. This time we only do one electron chain, but we let those electrons, we burn those electrons out, making our hydrogen ion gradient. And we go but this time who's going to collect it is something we need every second, every moment of every day. Oxygen. And oxygen collects, binds with, it's the final electron acceptor, and with a couple of hydrogens forms what? You know what? Water. It will form water. And is water one of the products of cellular respiration? Yes. Okay? So we're going to take the glucose, we're just doing the reverse. Remember how in photosynthesis we fixed CO2 into glucose? Okay? Now we're oxidizing the glucose and releasing what gas? CO2. It's reversed it. Remember before when we did our electron transport chain in photosynthesis and we did our ETC, we brought them in? We brought the electrons to the interior. Guess what we're going to do now? Push them out into the outer space. We're still in the mitochondria, but to the outermost compartment. Okay? So it's basically the opposite. So once you have photosynthesis down, just do it backwards and you've got cellular respiration. Yes?
So if all cells, including plant cells, do this, couldn't a plant cell that has both mitochondria and chloroplasts like perpetually keep itself alive?
Yes.
Like,
so what do you think? Riddle me this. Think it out. What do you think a plant does more? More cellular respiration or more photosynthesis, and why?
Photosynthesis.
It's got to do more what? Photosynthesis. Why?
Because we take respiration.
Because we're alive. I am currently breathing. Why am I breathing? Because a plant did enough photosynthesis that it released oxygen gas that it didn't need for cellular respiration, so now I can breathe. Plus I ate this morning.
So if I put a glass jar over a plant, could it like survive?
Yes.
Forever?
Yes. Now, if you put that glass jar, you might build up a pressure, right? Because you're generating all that oxygen gas. So you'd have to have a release valve there.
So it can like keep itself alive.
Exactly. Absolutely.
Why can't we do
Okay. But think this out, because we don't have any. What is it going to need a source of?
Sunlight?
It'll need sunlight, but what else does it need a source of? Carbon dioxide. So it's going to need carbon dioxide. So it could do it just even Steven, but it's going to need, you know, if it's going to do more, it's going to need some carbon dioxide. So what we're going to do is we're going to go through each of these steps and understand them. But you already have a great understanding of how to do it because you understand photosynthesis. Now take a look at your notes on your steps. And I didn't have you do a lot of writing on your notes. It's mostly about you flipping your pictures and highlighting in bold because I knew it would be the last couple days before the exam and I wanted to make sure we could move quickly through the material. Okay? So aerobic respiration, if you're going to do it aerobically, you're going to do glycolysis, which you see here. Okay? That's going to be step 1. Step 2 is called the preparatory reaction or transition. Step 3 is this citric acid cycle, or another name for it is Krebs cycle. Okay? And then step 4 is going to be your electron transport chain, which is connected with the chemiosmotic synthesis of ATP, connected as in coupled with. If you do anaerobic respiration, aerobic is with oxygen, anaerobic is without. You will do glycolysis here, okay? But your next step is just fermentation. And the only reason you do fermentation is to regenerate the reactants you need for glycolysis. And we'll talk our way through that. So let's start with glycolysis. And here is the Minecraft version of glycolysis. Now, this is a 10-step process. But in a nutshell, what you do in glycolysis is you start with a glucose molecule. And just like what did we do with water? We what? Break it in half. And when you're done, if you instead of having one C6 molecule, if I break it in half, what will I have? 2 C3 molecules as a result of that. And that end product is called pyruvic acid, or another name for it is pyruvate. Okay? And so you can see here's a C6, and now I have 2 C what? 3s. To get the party started, to make it break in half, you can imagine if you're going to break something, there's an energy investment in that, right? It doesn't just, you know. So I'm forcing the break. It costs me 2 ATP. Listen careful. Costs me 2 ATP to get this party started. So I'm outlaying ahead of time 2 ATP. That's an energy investment. But when all 10 steps are done, it's going to reward me with 4 ATP. So I spent 2. I got 4 back. What do I net? What do I walk away with? 2. I get to walk away with 2 ATP as a result of that. I also walk away with 2 pyruvic acids. Look what else I walk away with. Look at this picture. Now you know where the 2 ATP come from. What else do you walk away with? 2 reduced NAD. That is like a check that's getting weighted to, you know, I'm going to cash it, right? Cash it at the bank of the mitochondria, right, at the ETC. So I am oxidizing. I have started the process of oxidizing. And it's one of those things where you oxidize it, oxidize a little more, oxidize a little more. And each time you oxidize it, you're going to get a little more NAD. You're going to write a few more checks that you can cash out later. Okay, so I walk away with 2 reduced NAD. So let's learn the song, and then I'm going to show you some more details on this. Okay, so look to me, look to me. And again, you have to learn my parts too. What is this whole thing called? Cellular respiration. Come back to me. Look at me. Look at me. What is the whole thing called? Cellular respiration. And I'm going to ask you, who does it? You're going to go, all cells. Okay? Who does it? All cells. And I'll say, where do they do it? And you're going to go, in the cytoplasm. In the cytoplasm. Okay, what's the whole thing called? Cellular respiration. What's the first step called? Glycolysis. Say it with authority. What's the first step called? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. Then I'm going to ask you, what do you start out with? And you're going to tell me glucose. Do you see how I'm trying to make the glucose? You know what the monomer looks like, right? Okay, glucose. So just go like this. You can't be creative on this. You just have to do it my way. So do this. You can't go, oh, I'm going to, no, just do this. Okay? And then I'm going to say, what are we going to do? And you're going to tell me, break it in half. Okay? So what do we start out with? Glucose. What are we going to do? Break it in half. How many steps? 10. How many enzymes do you think we're going to need? 10. Okay? And what are we left with? And you're going to say 2, because you're holding them up. 2. And then you go pyruvic. Now, why did I go like this? There's 3 carbons in it. Yeah. Okay? So you're going to go 2 pyruvic, and remember, you're going to go acid. Okay. There's a little bit more to that. So let's start out with that. Okay, what is this whole thing called? Cellular respiration. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons in glucose? 6. How many carbons? 6. What are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. What are these called? 2 pyruvic acids. Okay, then take this hand right here. And you reduce some NAD, and you reduce some NAD, and make some ATP, and make some ATP. Then you go like this, 2. Okay, now why are we doing that? We made 2, but you know in your head, we spent 2, and we walked away with 4, so we netted out 2, and then you go right back here to your pyruvic acid. Okay, whole thing. What's the whole thing called? Cellular respiration. What's the first step called? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are you going to do? Break it in half. How many steps? 10. How many enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Perfect. Okay? Now, take a look right here. Now that you know that, this is really the 10 steps, and they're not even showing you all 10. You do not need to know the intermediates, okay? But so you can see the patterns between the 2, okay? I don't know if you can read that up there, but that says glucose. What are we investing right here? ATP. What did I tell you had to do to get the party started? Yeah, give 2 ATP. And then again, here we go with spending another ATP. Okay? When you are done with this intermediate, you know what you have? From our last reaction, what did I tell you made right here? G3P. This right here is G3P, or PGAL. Right here. So you can see when plants do photosynthesis, they don't necessarily take their 2G3P and synthesize a glucose with it. If they need to do cellular respiration, they'll just start right here. And if they start right here, then they don't have to spend 2 ATP to get the party started. Got it? Okay. Now, it's very tiny here. Let me see if I can, it's still going to be a little bit blurry here. Okay, there's your G3P. You can kind of make it out. Then do you see what are we doing right here? We're reducing some NAD. Reduce some NAD, and you're going to make 2. Okay, and then if we continue going out here, what are we making right here? 2 ATP. So now we're at 0, right? Because we spent 2, we got 2 back. Keep going, keep going, keep going. What did we make again? 2 ATP. Okay, and then we end up with 2 pyruvic acids as a result of that. Now, when you make this ATP, it's called substrate level phosphorylation, and it's basically a handoff in order to make your ATP. Okay, when you do that. So it's just one phosphate handed to another one. Right? So when you're done, you're here. Here are your inputs, here are your outputs. Not it.
Not it.
Pass or play, explaining inputs and outputs. Go ahead.
You have a glucose, 2 NAD, and ATP to get started, and then ADP and phosphate to which are which compose ATP, and then the output to get from it to pyruvate or pyruvic acid to reduced NAD to ADP, 4 ATP total, but then because you already put in 2 and you got 4, basically they're 2 gains.
All right. So now we are outside the mitochondria and we're doing glycolysis. It occurs in the cytoplasm by all cells. Good. By all cells. You break down glucose 10 steps into pyruvate. It is a universal reaction. It probably evolved first. You have an energy investment and activation of glucose using 2 ATP. Splits into 2 3-carbon molecules called G3P. And then you have energy harvest, 1, 2, 3, ping pong back and forth. Whoever did the explaining of this, you just do number 2, and other bio buddy, you do 1 and 3. Go. Okay, come back to me. Take a look at this part up here. This, if you have oxygen, you're going to do transition, prep, go on into the mitochondria, and crank out a bunch of ATP. But if you don't have any oxygen, your next step is going to be quite different. So here's glucose, investing 2 ATP up here, 4 ATP out, netting out 2. Here's your pyruvic acid. If you have oxygen, you go this direction if you have oxygen, and you're going to be in the what? Mitochondria. And you're going to do aerobic respiration. But if you have no oxygen, you're going to do fermentation. Now, depending on what kind of organism you are, will depend on what you do with your pyruvic acid. You and I, we will convert our pyruvic acid into lactic acid. And we're actually reducing it into lactic acid. What will lactic acid do to your muscles? Make you sore, right? Ultimately, it will make you sore. When are you sore? When you're working out and pushing yourself and you don't have enough what? Oxygen. Okay?
Didn't soreness happen after?
Yes, it does. Yes, the accumulation. Okay? So now, other organisms, when they do anaerobic respiration, they will make alcohol and CO2. So that is their byproduct. This is bakers depend on this, because yeast, you use yeast to make the CO2. What does CO2 do to the bread? Rise. Does Budweiser depend on this process? Yes, they do. Okay? Because this is how you generate alcohol. And beer, okay, different kinds of beer and wine depends on what you're fermenting. Are you fermenting a grain or are you fermenting a grape? If you're fermenting a grape, what are you going to get? Wine. Okay? You have a highly suggested reading and thinking on this one if you've already looked at it. Okay, so we'll pick up on the fermentation process next time. Okay, do we have a quiz next time? Yeah. Yes. Do you have basically all the notes, what you need? No. Yes. Okay. So the reason I'm telling you that is look them over. There's only a few spots that are missing so that you make sure you have it all good to go, because I'm going to see you one more time. When's the next day I'm going to see you? Thursday. And then what happens next Monday? A test. Yeah. Okay? All right. Make good choices.